Maravilha. Então eu vou começar aqui, tá, pessoal? Hi, everyone. So we're back with the webinar series. We're now having today Dr. Claudio Silva, my dear friend here from Uberlândia, from the immunology department. Claudio was formerly he did his undergraduate studies on odontology, but he's been since then working with host parasite interactions and immunology, and he has a lot of ideas out of the box. I think it's a good shift from the last talks we had with for him to present his ideas to us and to generate discussions. So, Claudio, thank you very much for being here today, and we're looking forward for your talk.
Thank you, Thiago. I'm very pleased for this invitation. It means a lot to me, this chance to share my conflicting ideas about science, about life, human being, and Homo sapiens, and the ecosystem. I'm very pleased. Thank you very much. Thank you all for listening, and let's try to rethink well, we think that we know until now, né? So this title, Decolonizing Host-Parasite Interactions for a New Era of Research, is concerning about my research in my lab, that I'm trying to shift some questions, not to change, but to add more points of view in the process of making science. And my inspiration for this shift is based on this writer, Chimamanda is a Nigerian writer. She had this talk in TED Talks, The Dangers of a Single Story, and this challenged my life from the first time I saw. I see her speech every single day of my life, I think, trying to get more from what is about a single story. Uh, the process of mental colonization is so dangerous because when I have listened Shimamanda along all these years, I have just thought about racism, prejudice against race. Nothing else troubled more than racism. But is there only a single story about human race? This is the only single story tell. When I got in touch with Malvina's song Little Boxes, it's a song much cute. I love it. And she's criticized the architecture of her time, 1967, but not only the architecture of houses that were building exactly the same one as other, but she make reflections about society, about university. If this thing about many years, what we do more than only fragmentation of knowledge, because fragmentation of knowledge is a way of putting students into boxes. So, uh, what else could we do more than the fragmentation of knowledge? Maybe it's the knowledge itself. It's a way of putting people into boxes. So I propose that uncertainties makes a move, makes a go to another path. So let's try to see other possibilities of our lives. Bear in mind that we are involved by single stories of lives, and we are just putting people into boxes, and university plays this role. So I don't want to rebuild or reshape the already much binary world that we have. We have good, evil, right, wrong. It's not about this. The question is not what is right, what is wrong, but the question that I want to propose is what you can add. Can we listen that we have not listened among centuries? So it's an invitation to see our lives, our world from outside of it. So let's see Earth from the space and our society from the space and what we see. We see an anthropocentric world, the human being, né, the Homo sapiens, that is not it's not sufficient to be the special species. Né? We have to introduce novel terms. So human, né? Human controls the world. So we have an anthropocentric view of the world that makes us the rights to destroy the plants, to destroy animals, because the world exists to serve us. And this is a point of view shared by kind of Homo sapiens group that have existed in this world. We are surely not. Né? Surely this anthropocentric point of view is not shared by all communities of human beings that existed. So this point of view was introduced and maintained, sustained, and kept by an Eurocentric point of view of the world, of civilization, of what is civilized, what is not civilized, what is primitive, what is right, what is wrong. They taught us to commemorate, to celebrate an invasion process, a genocide process. They told us all we have done, shared, and done up to now, we have not made it by ourselves. Our beings that occupies a place that was occupied by others. Né? We are a really out of space people. And science is not neutral place. Né? Science is influenced by the Eurocentric point of view, the conquest. Science begins, né, with the conquest of the world by Europeans. So we have an European point of view and the relationship between parasites, pathogens, and hosts is an European point of view established by Koch in the germ theory, where the human body was a sterile place that it was invaded by a microorganism, an invader and stranger, something that is not self of the body. And immunology comes together and turns our bodies as a place of conquest, uh, internal battle. Now we live an internal battle inside our bodies concerning what the point of view we have learned, have been taught and learned and propagating across generations. So we have ourself and ourself. We have one pathogen, one disease. So we have a point of view that made Descartes establish the modern science that we have not passed through it already. It's a reduction point of view that we can separate everything into small pieces. Understanding these small pieces, we can understand the complete function of our body. So this reduction is our main platform of experimental desires, designing one pathogen, one disease. And this one pathogen, one disease way of thinking and the colonialism way of doing they came and extract everything they want. They stopped it? No. They continue extracting from South disease, Americans, African disease to protect the North. We have reproducing all this, this behavior in our days. We extract from our volunteers in hospital. We collect their blood. We do not give much back of this of the experiment. Or we will we want them, the volunteers to sign the paper, and that's okay, let's do science. This is the way they always did here in Americas, and still do in Africa, and we do the same between us, among ourselves. We continue extracting, we continue establishing extension projects that we establish the problem, we have the solution, and we go to the community and give them the solution, and we have no uh, no no we have not tried to understand to see if that is a really problem for that community. So it's the reproducing way of thinking of the colonialism is still among us. We continue to do this between ourselves, our people. So the cats thought that we could separate things and understand the complete world. And in 1980, two French scientists coined the rhizome, a way of thinking science rather than the reductionism of Descartes. And the rhizome was the point of view that established systems biology that began in the 2000s, and I believe that it would shift our way of things. I was very, I myself was a student in Unifesp, and an enthusiastic of systems biology. I have bought all the books that were thrown to be were sailed at that time. Those books with those formulas so difficult that I've never read any of them. I have many of them in my house, but I couldn't read those systems biology book that promised science that would rupture, make a boundary science, and to leave a reductionist behind. But what we see is that we have sequenced everything we can, all the genome, transcriptome, metabolomes, genomes, and we keep on reducing things into algorithms. So we have put people inside our computers into small pieces in the kernel. And I think that the relation, the host pathogen interaction relationship is not the center of our questions any longer. It it was left behind. So, do you really rupture with reductionism? Systems biology really saved us? I don't know. And we going to a situation of precision medicine that we desire and design and develop medicines to direct a target, and it's okay. It's fun. It works. It's important to target what we have known. Um, concerning many kind of disease. But when we talk about parasite, especially parasite-host interaction, we are talking about poverty. Systems biology would really be the best way of solving poverty problems by personalizing a therapy that will be not afford the most people will never afford it. So I don't know. With thinking about precision medicine in host parasite pathogens interactions is the best way to try to solve all these remaining perpetuated problems because be sure that the projects that are being developed and has been developed in Africa, sustained by Bill and Melinda Gates Foundation, for example, uh, did not develop any university of Africa, did not, probably did not granted scientists living in Africa and African scientists as a PI we can share, we can see that this can be an experiment we can do in our computers by visiting these institutions that give lots of money to solve problems in poor countries. What these projects do to better the way of living in these countries instead of only extracting from them and publishing new articles in the literature. So, uh, United Nations is not naive. So one day, the 2000s, they have thought about the invaders. Now, what was the primary impression of the invaders that came across Americans, and they mentioned that the animals, the parrots, that the captain of the ship here in Brazil. So they remember that the relationship of indigenous people with the ecosystems is not the way we do. So we have established a unique and for the first time in human life, the one way of thinking that is really an invention of the, the, the society, the contemporary society. It's a joke. One health is something that existed uh, much longer than the invasion process. So living, thinking our health, the way we have place for it's not sufficient. We need to be concerned about the environment that we have the rights to destroy across these centuries. So the concept of One Health, the recognition of the ancestors' thought, the traditional thinkings, the traditional way of being the recognition, I think that it's not too late, but we can try to use this recognition for us in the South, in order to extract from us to save more problems. Uh, so the concept of it's not new. Nothing I'm talking about here is new. It's an invention of my uncertainty. It's not an invention of anyone. This is fragmented in literature wherever we go, whoever we read. So the concept of that we are not an stereo species, né, body that are invaded all the time by different dangerous bad pathogens. Our body is an oral biome, so we need to try to understand. Maybe I will try to understand the ecosystem involving the disease I want to give any contribution that could reach the one who is in susceptibility of that. So it's not being horizontal or vertical reductionist or non-reductionist science. We can do everything. So it's a call for ends to add, and not to remove or shift. It's to include new perspectives. So we do not have to change one way to follow. We have to go to the ancestors, I think, and put them inside our labs. I think we can uh, understand the needs of those in susceptibility of the disease of our challenge, and try to introduce their lives in our lab at the best way possible. So maybe we can shift from one target to another ecosystem targeting process, and maybe we can have additional and good results to help the solution of the problems we face. So we do not need to change to kill the parasite, to eradicate an agent, but we can restore the resilience of the organoids, the disease organ. We can try to understand the social situations and reproduce the social inequalities in our lab and understand their impacts in immunological and biological process systematically. So, and this is my first chance, the first chance I had to speak professionally about my ideas. That's not only mine. I know that, but it's it's conflicting, and people always says, Cláudio, you are a figure, né? Você é uma figura. I know that we have many figures everywhere, and this review I put in the same piece of sheet all the questions I think that contribute to chronic inflammation, and I'm really bothered by chronic systemic chronic inflammation. I am 52. Maybe I'm feeling the consequence of being chronically inflamed. So this is in my uh, in my radar. I am targeting chronic inflammation wherever I do nowadays. So in this revision, I discuss social problems, social conflicts, polarization, political polarization, and all whatever else that leads to chronic inflammation. And the publication of this review made me believe that I could be here and talking to you, that I would have this right to tell about the things that bothers me. So I work with Leishmania amazonensis and Trypanosoma cruzi, Leishmaniasis and Chagas disease. These parasites are trypanosomatids. There are parents, relationships, parentholds, uh, and they are Leishmania. We have many species. Trypanosoma cruzi, we have many strains, but only Trypanosoma cruzi species. We have subspecies, but I'm not talking about that. And Leishmania amazonensis is a species of Leishmania that cause epidermal leishmaniasis, tegumental leishmaniasis. So I have been working in a reduction way of thinking, trying to understand P21 protein from Trypanosoma cruzi all my life long. Né? I have spent many money with that. One day, some years ago, I I think that we make many mistakes, and one of my mistakes was to publicize in media, social media, some results that came to a journal, a television, a television presentation. So one person contacted me about my results towards the cure of Chagas disease. That made me crazy, and I stopped from that time on. I stopped promotion of whatever I do because we create a kind of expectation that we cannot really sustain. And pay attention project and results were performed on this way of thinking on this knowledge. I have proud of my work, but I would not do any of these papers again. I would make everything much more different. Um, and concerning leishmania amazonensis, I have dedicated and understand the role of MIC2 and vacuolar expansion of this species. And it seems that this protein is involved somehow in vacuolar expansion. And my new approach is trying to mix all these things up co-infections, mixed infections of different strains of Tecruzi, mixed infections of different strains of Tecruzi and Leishmania amazonensis and other Leishmania. And what we have seen is that a mixed infection is something completely different from a single infection, each one of the single infections. So what we probably see in nature in proper life. It's not what we do in lab. And when we do in lab, we see that the what we think individually will never correspond to what the infected person would require. We see here the modulation of macrophage phenotype by co-infections between T. cruzi and Leishmania amazonensis. And also we see that when we co-infected an animal with the cruise and Leishmania amazonensis, we have more inflammation in heart. So what Leishmania amazonensis that keep herself in the paw, what it contributes to a higher inflammation in the heart of the co-infected mice. So this higher inflammation is related also to higher parasitism of T. cruzi in heart of the co-infected mice. So when we put T. cruzi and Leishmania in the paw of the animal, both parasites at the same time, at the same place, we have a more severe infection, a tuberculosis heart infection and symptoms. So, how can the Leishmania parasites that is not visceral contribute to the shift of behavior of the same strain of the cruzi? So it's something really interesting that we are trying to understand and putting into other contexts that I'm gonna tell about it. This is the co-infections between strains of the cruzi, virulent and low virulent, as it was named recently Cruzium by an elegant review from a group of researchers. So Cruzium is something that we may find in T. infected people, different strains at the same place. So here we see that the higher infective strain shows more infection in macrophages, né? Raw macrophages. And when we try to understand the cytokine profile, we see that each strain to have a pro-inflammatory or a low inflammatory profile concerning the effectivity of the strain. But when these different infective strains are put together, we have different profile. It's not a superposition or a reduction of concerning the presence of one or another strain. It's a completely, it's it's another shift. It's another lens of the cytokine secretion. Also, we are 12, we are 6. Here, immunomodulatory cytokine, IL-10. So we have a different profile, and when we have co-infection. Nitric oxides, also we have different liberation process, and when the different strains are put together, we have a different sign of NO. So what I want to think about, uh, uh, it means is against the process of science, because we are always evaluated, the post-graduation program is evaluated, we need impact, we need higher impact publications, and we need to pay for these high impact publications, so we establish an industry of science, the industry of science that we are living at this moment. This industry does not care about the Shagazi person in Pará, does not care about the leishmania infected person anywhere here in Brazil. So, the why and for whom I make science. This is the first question I am trying to make, uh, considering that I need to form scientists that can be competitive in the world of an Eurocentric point of view. So my students need to get uh, technical support. They need to learn technicians, but they need to try to make science for the infected people, and not only to form a professional. So, who will be the beneficiaries of my research done in my lab? My students will be get the graduation. I will be very much happy to occupy my time doing science, because what else could I do instead of working along the this is, in my life, this is the contribution to occupy my hours of day. This is how I feel about science nowadays. Science is in Brazil is so difficult to do, and we find a lot that we have such different feelings, that my current feelings about science is that it makes me live. It makes me live my day and be something. But I want to do something that may be somehow useful for one who claims for some changes. So, what is the scenario of Chagas disease? Where do these people live? They are have nutritional stress. They are co-infected with what? They are exposed to agrotoxicals. What about the microbiome in the intestine? We have brain intestine axis that is really important for chronic infection, and pathogens is another branch of sustained chronic exposure to be a systemic chronic infection. So, microbiota, pathogens, diet, nutrition, exposition to UV, any kind of compounds that are made every single day. We have to be exposed to a new compound, so maybe we can try to mimic in the lab what the person that is under possibility to be infected with Trypanosoma cruzi, maybe we can see in the lab mimicry what happens to him. And I think that if I try this, I will occupy my hours of day in a good thing, not just occupy the hours of my days of the rest of my life. Maybe this could be really a good way of occupying the hours of my day. Trying to understand studying the phytotherapies that I have was crucified. I never liked to study medicinal plants, potentials. I have always claimed against it. So I'm giving my face to be, and I'm gonna I'm already including this in my life, but I wanna know uh, the, the, the source of this therapeutic point of view. Where does it come from? We can go back in time, analyzing oral story of people, and we will know that different plants have different origins of traditional knowledge, including European colonists gathering with indigenous rural people and black people in Brazil. They defined that the Matheus e Cogli had importance to paint. So the I think we could, we need to put in our publications the story of that plant, the people behind that scene. I think this would make it will not hurt us, and it will make a big difference in the way in the movement to change the way we do science, to fight against the, the single story of our lives. We have been living a single story dictated by someone who does not live among us. So I think that we could try to rethink the way we can direct our lives. And I thank you very much.
Okay. Thank you, Claudio.
Muito obrigado.
Uh, okay, questions. Usually the students will go first. Uh, someone in Rio, São Paulo. If no one has it, I just had want to continue provoking you, Claudio. So you thought you said you're gonna do stuff or talk about stuff outside of the box. Yeah, for sure you did, and I just want to understand how can we think outside of the box if we need to be inside those boxes, right? So, to ask for grants, to publish papers. They're the things, common sense that delimits what we do and what we present. And how do you feel about that? How can we, try to help effectively help people that are going through those neglected diseases or whatever, but still be inside the boxes science puts us. How do you think we can manage that?
Yeah, it's a kind of way of being, eating from all the sides of the cheese. What it means, it needs to be silent. It needs to be transgression. A transgression, maybe a transgression. What is being transgressed does not necessarily understand the transgression. And I think that that is the only way we can do this process. We cannot. stop doing, stop uh, giving foods to the industry. Uh, we have nowhere to go. We need to keep inside the box, but disrupt and subvert as long as we can. And I think that's the way I'm trying to do. This uh, this moment in my life here is one transgression is important. This is the first time I am talking to someone officially and professionally about the way I wanna do the job. And this is a transgression that you made it possible to me. So I think that we can get these opportunities along our lives. We can. We can write the project, write the manuscript, and publish, even though paying those thousands of dollars, we can do something inside all this. And more importantly, I think that um, the human contribution, the formation of our students, they need to know that they could think differently. Né? Uh,
Okay. Miriam, do you guys have questions in real?
Hi, yes, Thiago. So thank you, Professor Claudio. We have a student of ours here, Wendell, who has a question for you. Just ask him to come and make the question. Thank you again for your talk.
And I want to first thank you for the presentation. I really do think the presentation that you gave, the subject that you spoke of was really interesting. And I have a question because I really like the way that you spoke about different things, how we should actually change the way we do the research. But then I have a question. Uh, the scientific community in a whole is very solidified, in the way they do the research, right? We all have our own workflows that we follow, and we've been following for years and years now, right? And how do you think we could break this down and change a little bit the way we do the research and a little bit of the topics that you spoke of on today's presentation?
yeah, I think that this will be a process. Uh, I think that this moment we are having now, it's really important to our lives, for mine, for yours as a young scientist, and we cannot do this at once. Right now, I'm gonna rupture with everything. No, it's not possible. We live under the Ecuador. So we are in the south of the world. No, we are under, né? Under, you know. So there are many people above us. It's not possible to make a rupture uh, at once. We need to progress. I think that we can change our vocabulary, it will be interesting. We can talk, for example, why do you insist to tell that parasites invade our body? It's an encounter, they meet each other, but the relation is not, there was no synchrony among this conversation, the parasite and the host. So we have an infection, but it's not an invasion. It's not this is not a bellic, it's not a war that it's uh, in our body. If we have a war, thus do we tolerate the lives, such bacteria of the microbiota. So it's not its interaction is not an invasion. So if we begin to change the vocabulary, if we begin to change to problematize, to question. to whom I'm working. I'm working for me. I'm working for really. I'm working for camps, or I'm working for the
Essa
people
pergunta aqui.
that people in the country. So we can do all of this, but we can do more for the people in the country, pretending we are serving camps, pretending we are serving MEC and Wiley and Elsevier. So we cannot only serve, and we can tell them, I'm serving you, but I know what you're doing. But instead of only serving you, you need to swallow these people that I want to solve this problem. So we can make them chew and swallow the ones we are concerned about. We can put our life problem, the people we need to help in the world, and put them, insert them in our boxes in the university, in our Cartesian or whatever it be way of doing. We need to exercise the possibility of really introduce them in our way of doing, and try to forget the parent, né? The external body, the isogenic mice. This is not that. This reflects. This is nothing concerning parasite, pathogen host parasite interactions. What we do reflects nothing at all, because it's not the real world, né? It's a kind of playing games, unfortunately.
All right, thank you.
Thank you for the question. Anyone else? This question for Professor Claudio, São Paulo, Miriam.
Thank you.
There are no questions here too, Tiago.
Okay. If we don't have any further questions, Claudio, thank you very much for your time, your ideas, and thank you very much for everyone for being here. Until next week, okay?
Muito
Bye bye.
obrigado, gente. Valeu mesmo. Obrigado.
Obrigado.
Obrigado,
Tchau,
gente.
tchau.
Até mais. Tchau,
Tchau.
tchau.
Obrigado. Tchau.
Acabou. Mandou?